we, as a clinician that work in the field of MS, always think in terms of the correlation between gray matter atrophy and accumulation of disability. But first, this correlation is not that strong. And second, we do not really know what is driving this correlation. In the field of MS, we underappreciated the role of synapses, in particular when we compare our field with other fields like the Alzheimer's disease one. And very importantly, we do not know which is the compartment of the neuron that could be cell body, axon, dendrites, but also synapses that is first affected by demyelination. And so to answer all these questions, we took an approach that went from a translational approach, working with EAE, to look at OCT, so retinal imaging in our MS patients, to then look at proteomics to try to understand which is the mechanism that drives synaptic loss. To answer this question, we have to think about the complexity of a single neuron. A neuron has a cell body, a single axon, dozens of dendrites, and an average of 7,000-8,000 of different synapses. So if we put the complexity of this single cell within the complexity of our CNS, it will be extremely complicated, and I would say impossible, to use a tissue-specific imaging to look at synapses. We will always need immunohistochemistry to do that. But the retina is the perfect area of the CNS where we can look. Because if we think about a cross-section of the retina, the first layer, the top layer, will be formed just by axons that go to form the optic nerve. Beneath it, we have a layer that contains the cell bodies of the retinal ganglion cells. And beneath it, we have this layer that is containing the connections between the retinal ganglion cells and the bipolar cells. And the retina has a second advantage that is amazingly interesting when we look at demyelination. So demyelination can happen only after the lamina cribrosa in those same axons that form the first layer that we were saying before, that go through the lamina cribrosa and go to form the optic nerve. So basically we can see in the retina the changes on a circuit level as a consequence of demyelination that is happening downstream in the optic nerve. The first part, that is the part associated with the work on EAE, was based on the fact that we wanted to understand which is the first compartment that is suffering as a consequence of demyelination. So we looked at our mice in our lab. Mice develop the first symptom around day 12, then they have a peak of disease around day 18, and then they develop a chronic disease. So I collected the retinas of these mice at these time points, first day of symptoms, peak of disease, and chronic stage. And we looked at the IPL. The IPL is a layer in the retina that contains synapses. We already had a lot of information about when we can see the loss of the retinal ganglion cell bodies. And looking at the IPL, thanks to the work also of another lab, Dori Scheffer lab, and in particular Sebastian Wernenburg, that did the staining for synapses, we saw quantifying the actual number of functional synapses, so multiplying the density for the volume of the layer, we saw that there was already synaptic loss 12 days after immunization, so even before the first symptom. And very importantly, we saw that synaptic loss was increasing with time and was more and more, and in day 60 it was a lot more than at day 12. At this point there was a part of our work that was to develop the IPL thickness in the OCT as a measure that is reliable and reproducible. And so for doing this, we did a retest on 20 healthy subjects. This was very important because previous technologies weren't able to dissect out the IPL from the GCL. And we found out, looking at different metrics, that our layer was doing well and even better than other measures that we commonly use in our research. And then with this in mind, we went to look at our patients. So remember, we found out that synaptic loss is increasing when the disease becomes chronic. So we decided to look at patients that do not have any more relapses, but start accumulating disability. So this is what we always call the transition to secondary progressive disease. Now our definitions are a little bit changing, but it is still a concept that is clinically meaningful. So we looked at the time before the transition to progression in people that were having a longitudinal follow-up with OCT, and we compared these people with patients that stayed relapsing remitting, but were very similar under a lot of points of view. So they were having similar disease duration and similar sex and age. And we looked at the annualized rate of change of all the layers, with the idea, of course, the hypothesis that the IPL was going to tell us something important. And what we found is that the only difference that was substantial was in the IPL. So people that became secondary progressive had a loss of IPL. And very interestingly, the only other layer that had a similar behavior was the OPL, that is another layer containing synapses, but between bipolar cells and photoreceptors. And I do not remember if before I told you, but the IPL contains the synapses between the retinal ganglion cells and the bipolar cells. So with this data, we were having just information about the timing of synaptic loss, and we wanted to try to understand which was the mechanism underlying these changes. So we took this proteomic approach using a proteomic tool that is called O-Link, and we looked at a protein that is a marker of synaptic loss, SNAP25, and we correlated it with targeted proteins that are markers of CNS injury and markers of inflammation. And we found that the correlation between synaptic loss and MOG was incredibly important, and it was staying stable even taking into account neurofilaments. So this meant that the demyelination is inducing synaptic loss independently on the effect of demyelination on axons. We found some other associations that were way less important than the one with MOG, and we didn't find any association with markers of inflammation, microglia, T and B cells. In the past, we have had some amazing work made by different scientists. In particular, I want to say the name of Beth Stevens and Dory Schaffer, that looked at synaptic loss, and they made it clear that synaptic loss is key in a great amount of neurodegenerative diseases. And their experiments were focused on looking at the role of microglia in synaptic loss, and they found out that microglia seems to be associated with synaptic loss in these diseases. With our work, we are suggesting a different process that is inducing it. So we are suggesting that it is actually demyelination, just in the field of neurodegeneration induced by demyelination, that is inducing synaptic loss. But of course, we are interested in looking more deeply in this process. We still do not have a molecular mechanism, and we submitted a grant to look at it. Another important point that I would like to stress is that we found out that the synaptic loss is associated with disease progression. And so we are interested in understanding if IPL could be a predictor of disease progression. And the last point that I would like to stress is, as I already said, that synaptic loss seems to be induced by demyelination independently on its effect on the axon. We are looking forward to use the IPL at a single patient level, even if so far we just did an analysis on a population level. Of course, to be able to do this, we will need to have more data, and in fact we are collaborating with another university to see if we can develop a threshold to see if patients under a threshold will be at higher risk to develop progression. And we are really interested in the IPL also because the retina is a layer where myelin is not present. So we are able to see the consequences of demyelination that is happening downstream. So what we are looking at is a change and on a circuit level in an area that doesn't contain myelin.